In this video, we will discuss about why aged CD8+T cells cause pro-aging, and that we have seen in the when we are discussing the results in the previous figure. So, in order to check the reason behind these aged CD8+T cells causing pro-aging, they plan their experiment that is shown in this figure A. So, how they come up to this experiment, and that from the literature they are able to know that they know that the CD8+T cells plays a major role with this G protein coupled receptors. So what it decided to treat these CD8+T cells with GPCR inhibitors, that is G protein coupled receptors inhibitors, before adoptive transfer into the young mice. And these are the different conditions that they have used. That is, they use the young CD8+T cells plus vehicle, and this aged CD8+T cells plus vehicle, and the aged CD8+T cells plus PTX. PTX is the GPCR inhibitors. You can get the full form of this PTX in the description. So they first do this CD8 plus depleting antibodies, and at day 0 they inject this young CD8+T cells and these other conditions, once depleting the antibodies that are present within the mice. And after that they did these studies, that is novel object recognition, and this RAWM, and that those results also we have discussed in the previous video without this PTX. And after that day 24 they analyze the CD8+T cells at different regions. So in the lymph node they are able to see a significant decrease in the transfer of CD8+T cells from the aged CD8+T cells when it is treated with this PTX. However, for the spleen and for the brain they do not see any significant changes, but for the blood they see an increase in this CD8+T cells, CD8+T cells when they are treated with PTX. The brain results are not shown here, but in the text it is mentioned. And for this overall, this aged CD8+T cells from the origin of the cells, it is CD45.2, they do not see any significant changes in the hippocampus, and this indicates that this overall aged CD8+T cells does not get affected by this PTX. Overall CD8+T cells are present in the similar amount as what they have injected, so that the comparison results is because of this inhibitions of the GPCR treatment. Afterwards they check the CD8+T cells at different tissue sections, and one of the representative image of this PTX treated aged CD8+T cells mice is shown here. Here we can see that the CD8+T cells are very low in amount, and when they check the total count there is no difference in the total count, only the difference that now they can see is the distributions. So in the vascular also they do not see any significant difference between the young and aged CD8+T cells, but they see a difference between the aged CD8+T cells and aged CD8+T cells with PTX treatment. And same thing they have seen they do not see any significant difference for any of the conditions. That is, total CD8+T cells there is no changes, vascular CD8+T cells no changes, and perivascular CD8+T cells also no changes, no significant changes, and this parenchymas, near parenchymas also CD8+T cells there is no changes. The only changes that they are seeing is in the lymph node, and also its counteractive is in the blood. That is, in the lymph node they are able to see the CD8+T cells in the PTX treated, aged CD8+T cells in the PTX treated conditions significantly reduced, and whereas there is increase in the CD8+T cells in the blood circulations when they are treated with PTX. This is what they have found out so far. So this is from that one can come to a conclusion or come to a inference that Aged CD8+T cells might interact with the GPCR for this lymph node transfer. This is what they come up from the results that are presented in this figure. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. And we will discuss the further figures about this, further panels about this figure 3 in the subsequent videos. That's all for this video. Thank you for your time.